Welcome to today's webinar. It is my great pleasure to host Dr. David Forrester, Senior Research Scientist at CSIRO in Australia. He will talk about modeling species mixtures and uneven-aged forests. We will start with a presentation by David and then followed by a question and answer period, and we should be able to wrap up this sort of official part of the webinar at 4 p.m. And sometimes we stick around a bit longer if there are still questions after that didn't fit within the time frame. Before I hand over the floor to David, I would like to acknowledge that I'm hosting this webinar from my UBC office that is located on the traditional ancestral unceded territory of the Musqueam First Nation. The land UBC is situated on has always been a place of learning for the Musqueam, who for millennia have passed on their culture, history, and traditions from one generation to the next on this site. Second, I would like to remind everyone that the webinar is being recorded and will be posted on the IUFRO webpage, hopefully by next week. And then third, I would like to acknowledge that this webinar is part of the 2023 Forest Mensuration and Modelling Chats, which are organized by IUFRO Research Group 401 Forest Mensuration and Modeling. If you have speaker suggestions, I'm still looking for speakers for September and November. Please email me. And I would also like to mention IUFRO's mission, which is to advance research excellence and knowledge sharing and to foster development of science-based solutions to forest-related challenges for the benefit of forests and people worldwide. And that the webinar series is being organized in the spirit of this knowledge sharing and to allow for some interaction and networking across the globe. And I was going to talk about the upcoming webinars at the end, so I will stop sharing. And David, the floor is yours. And thanks everybody for joining so far.
Thanks. Okay, so you should be able to see the screen, I think. So thanks for the introduction. It's pretty well known, I think, that sometimes mixtures of tree species can be more productive than monocultures, but sometimes they're less productive. And it's still quite difficult to predict which mixtures might be more productive and in which conditions they might be more productive. And this seems to prevent the use of mixtures in practice as well. So firstly, I'm going to talk about some of the species mixing patterns that have been found in the literature, and also some of the patterns relating to how size diversity influences productivity. Then secondly, I'm going to talk about some of the processes that influence those patterns. And thirdly, about some of the modeling work that we did to try to summarize those patterns and processes. And in particular, we were interested in developing a model that was simple enough that it could be used by practitioners as well as researchers. This graph shows many different examples of the relationship between tree species richness on the x-axis and then productivity on the y-axis. And here the productivity is expressed as the productivity of the mixtures relative to the monocultures. So anything above 1 means that the mixtures were more productive than the monocultures. Averaged across many different studies and sites, the mean mixing effects are normally around about 20 or 30%. But you can see that there's also some cases where there's no effect, so they're along that 1 line, or sometimes there's even a negative mixing effect. There's a lot of information in each of these lines. So to be able to model this, it's useful to break it down a little bit. And there's 2 main points that are worth considering. The first one is that each of those lines is the mean of many different points. And often there are going to be some very efficient species whose monocultures are actually more productive than some of the most diverse species mixtures. And we wanted the model to be able to capture some of this variability so that we could predict when this is going to occur. And the second point is that this increasing this pattern of increasing productivity with increasing tree species richness is interesting, but it's not so useful when trying to understand the functioning or the management of mixtures. And that's because any given combination of species can interact differently on different sites. So the mixing effect might be 20% at one site, then 40% at another site, and then 0% at another site. And this is still quite difficult to predict. And it might also be one of the reasons why nearly all of the world's plantations are monospecific. So the cause of this variability needs to be understood if we're going to be able to model it. So this slide tries to explain some of this spatial and temporal variability in mixing effects. The y-axis is showing the mixing effect. So up the top, the species would be larger in the mixture, and down the bottom, it would be the same size or less in the mixture than in the monoculture. Then the x-axis shows any spatial or temporal gradient in growing conditions. And as an example, I'm just going from low nutrient availability on the left up to high nutrient availability on the right. And then the blue line shows what we might expect if the species interactions improve nutrient availability. So if there's a higher nutrient availability due to species interactions, we might expect that that causes large mixing effects on low nutrient sites, but on sites where there's already plenty of nutrients, then adding more nutrients by nitrogen fixation. For example, in this mixture with Eucalyptus and Acacias, it's probably not going to have much of an effect. In contrast, if the species interactions improve light absorption, then we might expect that this is more useful on a very nutrient-rich and moist site. So, for example, in these spruce and fir mixtures, on a site that has lots of nutrients and water, we would probably expect that there's high leaf area indices, and so intense competition for light. So interactions that improve light absorption or light use efficiency could lead to large mixing effects. But on low nutrient sites, there might not be any mixing effects at all because it's the nutrients or maybe something else that's limiting to growth. Most of the studies in the literature seem to fit within this relatively simple framework, so it's a useful first step. However, to make use of this information, it's important to know which processes and interactions are actually occurring within the mixture. And this slide shows many of the different processes that have been found to cause mixing effects in forests. And I'm not going to go through all of them, just to mention that some of them are related to nutrients, some of them are related to water, some to light, and then a few others don't really fit so easily into these categories. Some of these processes will have bigger effects on some sites than other sites, and their effects can change in importance through time as well. So when interpreting the importance of these different processes, it's useful to use the production ecology approach. For example, to use the production ecology equation. And this is a very simple equation where the productivity or the growth is described as a function of the supply or the availability of resources, the proportion of resources that are captured, and the efficiency with which those resources are used. And this quite simple equation was developed more than 40 years ago, and it was developed for agricultural monocultures. But it's also useful for examining mixtures because it can be used to show how the different species are using the different resources. So how the resources are being partitioned to the different species and how efficiently those species are using those resources within the mixture. As an example of this production ecology approach, this is a mixture of Eucalyptus globulus in the upper canopy and Acacia mearnsii, which is in the lower canopy. And the acacias are nitrogen-fixing species. And this is actually a combination of species that occurs naturally together, but this particular photo is from a planted experiment. So the objective of the experiment was to maximize biomass and wood production while minimizing synthetic nitrogen fertilizer application. And this graph shows the volume that was produced on the y-axis and the different treatments on the x-axis, where the number represents the percentage of that species in the treatment. So on the left is the 100E, the Eucalyptus monoculture. On the right is the Acacia monoculture. And in the middle are the mixtures. And so the mixtures were more productive. So presumably there are some kind of species interactions that were causing this increase in productivity. So to understand what was going on, the production ecology approach was used. So firstly, there were relatively high rates of nitrogen fixation in the mixtures, but there were also much higher rates of nitrogen and phosphorus cycling through the mixtures than in the monoculture of the eucalyptus. The mixtures were more productive, so they also used more water, but they were also much more water use efficient than the monocultures. There were changes in crown architecture and gas exchange, and there were also increases in the light absorption in the mixtures. And in addition, there were higher light use efficiencies in the mixtures than in the monocultures as well. And probably because there were higher nutrient availabilities within the soil, the trees changed their carbon partitioning. So they shifted their carbon allocation from below ground more to above ground to capture more light and fix more carbon. So this can be described using the production ecology equation. And if we just take the eucalypts as an example, we know that the eucalyptus trees in the mixtures were more productive than in the monocultures because the supply of nitrogen and phosphorus increased. They captured a higher proportion of the water and the light, and they used all of these resources more efficiently. And this was caused by structural changes and physiological changes. Often, many of these different processes were occurring simultaneously, and their influence probably changed through time. So it's important that the model can replicate this type of spatial and temporal dynamic. Another point is that many of these different processes don't actually require species interactions. They can also occur in a monoculture. So these bold interactions here, they can occur just because there are differences in size. They don't actually need different species. So this is the graph that I showed earlier about the relationship between tree species, diversity, and productivity. This is a similar graph, but this is now showing size diversity in terms of the diameter variability or the height variability. And the y-axis is the productivity of the size diverse plots relative to the least size diverse plots. And you can see that in some cases, there's a positive correlation. In some cases, there's a negative correlation. And this negative correlation was attracting a lot of attention because it was also occurring in plantations. And one example is these very productive eucalyptus plantations in Brazil. And these are clonal plantations. So the variability, the size variability, is probably relatively low here compared to other plantations and native forests. But even with that size variability, they were finding that they had 10 to 20% reductions in productivity. And this type of negative effect of size diversity on productivity has been found in other types of forests and plantations around the world. But curiously, there are also examples where there's increases in productivity with increase in tree size diversity. So to model and understand what's causing this size diversity effect, it's worth breaking it down a little bit to work out what's actually causing the size diversity in the first place. And there seem to be two main factors. The first one is the shape of the relationship between individual tree size and individual tree growth. So for example, whether that relationship is linear or whether it's exponential, which would mean that the larger trees are growing more relative to their size than the smaller trees, or whether it's shaped like the one on the right, where the smaller trees are actually growing more relative to their size than the larger trees. And the second factor is the shape of the size distributions. These two factors seem to interact with each other to influence the size diversity. For example, this black size distribution down the bottom would end up going to the red size distribution if the relationship between size and growth is linear. But if that shape of that size growth relationship changes, then the size distribution that will come from that black distribution will also change. And so this means that as the size distributions become broader or narrower or more or less skewed, then this is influencing the size diversity. And this will also be modified by mortality and thinning and recruitment. But the main point is that it appears as though the apparent influence of size diversity on growth might really be a side effect. It looks like the size diversity is really correlated with these two factors, and it's these factors that influence growth. It's not really the size diversity itself. And this can be shown a little bit differently in this slide. So along the top is different shapes of the relationship between tree size and tree growth. And then down on the left is different shapes of size distributions. And then these numbers show the productivity, the basal area growth, the meter squared per hectare of the different combinations of size growth relationships and size distributions. And all of these are simulated stands with exactly the same stand density. They all have 40 meters squared per hectare of basal area, and they also have exactly the same shape of the size distribution, but the growth of the stand can vary a lot. And the reason why is that in this particular stand, a high proportion of the trees are in the middle size classes. So we would expect that stands that have fast growth rates of these middle size classes are likely to be the most productive. So that would mean this stand, second from the right, would be one of the more productive stands, whereas the ones with the low growth rates of those middle size classes, which is the one all the way on the right, would have a much lower productivity. And similarly, if we have the same stand density and the same size growth relationship, the productivity can differ just because there are differences in size distributions. So for example, if we have the left-hand size growth relationship, the fastest growing trees are the largest trees, of course. So if we have stands where a high proportion of the stand is in large trees, then we would have the highest growth. So this framework helps to explain why size diversity has inconsistent effects on productivity. And it's really because of these two factors and how they interact with each other that can cause those positive or negative correlations between size diversity and productivity. And so size diversity seems to be more of a side effect. And because of this, we considered that it wasn't so important that we have size diversity in our model. It would be probably okay if we just have a small number of size classes. So the rest of this talk is about how we use the model to try to summarize and convert this information about these processes and the patterns into a form that can be used as a management tool. There are probably dozens, maybe hundreds of different forest growth models available. But for this purpose, we needed models that could reproduce those patterns that I've shown in the previous slides. And it also needed to be simple enough to have the potential that it could actually be used by practitioners and not just by us, by researchers. The next question was whether or not to use an empirical model or a process-based model. We've used all of these models in the past, and they're all very useful depending on the research question. Empirical models are particularly good if we have the data available, but for this project, often we were interested in new combinations of species or new management, or new climatic conditions. And so as a result, we generally didn't have the data that we would require to make empirical models. So we decided to use process-based models for this project. The next question was whether or not to use stand level or tree level models. And we spent a lot of time considering whether to use stand level or tree level models. We expected that tree level models would be required in order to model the interactions between species and between different sizes, and also to account for the vertical and the horizontal variability within the stand. And we spent a lot of time using detailed tree level process-based models, or doing analyses of inventory data and using competition indices, for example. But after all of that work, we actually decided to use a stand level model. So this was a bit surprising, and I'll explain why in a minute after I've shown the stand level model. And the other criterion was that the model needed to be robust. It needed to work across any different forest type and structure. And it also needed to be relatively easy to parameterize. So for this project and for the rest of the presentation, I'll be focusing on stand level process-based models. And this is certainly not to say that stand level or process-based models are better than other models. It's just to say that for the particular objectives of this project of modeling species interactions and this stand structural effects, we found that stand level process-based models seem to be better for this purpose. So the model that we used in the end was the 3PG model. And we used this model because it's relatively simple for a process-based model, and it's transparent, so we can easily look through the code. It's not so complicated. It's freely available as an R package, and it's been used and performed quite well in many different forest types around the world. And probably for these reasons, it's also used by some of the largest plantation companies in the world, and also to predict carbon stocks and water balances, and so on. 3PG also models most of the processes that we're interested in. The red ticks shown here are automatically included in the model. The yellow ticks are simulated as well. For example, if we change some of the parameter values through time. And that one with the orange cross, the shared mycorrhizal networks, isn't included in the model. 3PG is a stand level model with a 1 monthly temporal resolution. It has these 5 different main submodels. So there's a light absorption submodel, then that is converted into net primary production, which is partitioned to different biomass components. There's a water balance and mortality submodel. Then the inputs are some weather information, some site characteristics, some initial stand conditions, and species-specific parameter sets. It does the typical kind of management calculations, and then the outputs are the normal kind of mensuration data, and also some physiological outputs to help interpret that information. And importantly for our purposes, each of these different submodels had been validated in many different types of forests by different research groups. And so by validation, I don't mean comparing growth or stocks or mortality against observations. I mean comparing the outputs of a specific submodel, for example, the soil moisture or the transpiration or the light absorption, with observations of transpiration and light absorption. The 3PG model was originally designed for monospecific stands and evergreen stands and even age stands. So we needed to convert it into a mixed species model. And the biggest change that was required was related to the light calculations. The light calculations of the original model were that the fraction of photosynthetically active radiation intercepted was a function of the leaf area index and the light extinction coefficient of the stand. And so this is based on an assumption that the canopy is a single big leaf, a single layer with a single species. So like these simple diagrams. But we needed it to work for a much broader range of canopy structures and crown architectures. And so this equation was modified, but still quite similar to the original one. So it's still a function of the light extinction coefficients and the leaf area index of all the species within the layer, but it also has the vertical and the horizontal structure of the canopy accounted for. And this was validated by comparing the predictions from the 3PG light calculation equation with a much more detailed tree-level process-based model called Maestra. And the Maestra model has been validated in different types of mixtures and monocultures around the world, so we know that it works okay. And we found that the predictions and observations, or the Maestra predictions versus 3PD predictions, compare relatively well, but the Maestra is still much more accurate. However, the data shown here is from a very broad range of stand structures in terms of the vertical stratification of the stands and the crown architectures and the latitudes and the stand densities. And so although Maestra is more accurate, the 3PG calculations here are much simpler. There are far fewer parameters. It's easier to parameterize, and it's much faster to run as well. And because this means that the vertical structure of the canopy is included, it means that 3PG can also account for vertical gradients in vapor pressure deficit and radiation and aerodynamic conductance. So this influences the way species interact for water as well. The other submodels are all using relatively well-established and older approaches. So I won't go into the same detail for them, just to mention that the net primary productivity submodel is based on the maximum potential light use efficiency of each given species, and then that's reduced according to limitations caused by temperature or precipitation or soil moisture or CO2 or fertility. Then the biomass allocation to stems and roots and foliage depends on the soil moisture, the soil fertility, the tree size, and the vapor pressure deficit. The water balance is based on the Penman-Monteith equation, and the mortality is the simple self-thinning line, like that developed by Yoda. So the first example of using this 3PG model is from some subtropical forests in China. And they were interested in knowing whether mixed species plantations could be more productive than monocultures. And if they are more productive, then on which types of sites and with which types of management are they more productive? And the problem was that the most valuable species were always in monospecific plantations. There were very few examples where they'd been accidentally mixed with other species. So we used the monocultures to calibrate the model, and then the model was validated by testing it against the few mixed species plots that were available. And this graph shows some of the model performance. It's showing the observed basal area on the x-axis and the predicted basal area on the y-axis. On the left is the monocultures and on the right is the mixtures. And also for these simulations, the model was started at age 0. So that means that it was forced to simulate all of the temporal dynamics in the species interactions in order to get to this point. So it seems as though the model was performing okay. And so the next step was to use the model to examine different spatial and temporal dynamics of the species interactions. So this graph shows a soil fertility gradient, which is on the x-axis, and the y-axis shows the mixing effect. So it's the growth of the species in the mixtures relative to the monocultures. So anything above 1 means that that species were larger in the mixture, and if it's below 1, it means that it was larger in the monoculture. And just taking one particular example, this one with liquid amber mixed with Castinopsis sclerophylla, it was predicted that liquid amber would actually grow better in the mixtures on low fertility, but it would grow better in the monocultures on high fertility. And we also looked at gradients in water availability of the soil or age or stand density. And so the model was able to give a first impression of which combinations of species might be more productive in mixtures and under which conditions they might be more productive. And in these types of mixtures, most of the interactions were related to light absorption. Then the second example was to examine how beech and pine interact across gradients in Europe. In this case, there was already a lot of growth and yield data at each of these sites. So there were 26 sites, and at each site there was a monoculture of each species and a mixture of those species. And the aim of setting up that network was not to calibrate a model like 3PG, it was actually to examine the spatial changes in species interactions using statistical analyses of the inventory data. But even though there were so many different sites and plots, so 78 plots, and there was more than a decade of data for each of those plots, the spatial dynamics were still a little bit difficult to look at accurately. There was still a lot of variability in those spatial dynamics, possibly because there were several different gradients occurring at the same time. So soil moisture, soil fertility, and temperature. So as an alternative, the 3PG model was used. It was parameterized mostly using data from the literature and also some of the crown architecture data from the actual plots. And this slide shows images of the actual tree dimensions and the actual spatial positions of the trees that were measured in those plots. So you can see that there were many different crown architectures and canopy structures, the vertical structure, and also differences in stand densities. So this was a good test of whether the model could reproduce this kind of interactions. So firstly, the mixing effects were compared. So this graph shows the predicted mixing effect against the observed mixing effect. So the mixing effect is the stem mass of the mixtures relative to the monocultures. And this looked reasonably good. It's not quite as good if we compare foliage mass or root mass compared with stem mass. And then the model was used to look at different spatial and temporal dynamics of the mixing effects. And I'm not going to go through all of these, but one of the more interesting and probably useful results was that the patterns of species interactions that the model was predicting matched with other studies that had been done looking at the same species combination along the same types of gradients. So it seemed as though it was reproducing the actual species interactions. And the last case study I'll show is from these Swiss planter forests, these single tree selection forests. These forests are mixed species and they're uneven aged. So they have relatively complex vertical structures. And the size distributions look like that little graph in the bottom right. So they have these negative exponential shaped size distributions. And some of these plots have been measured for more than 100 years. So it was a good test of how 3PG can perform over a longer time span than in the previous case studies. So this was simulated by dividing each of the species into 3 different size cohorts. So each of the cohorts had the same basal area at the start of the simulation. And then the actual thinning treatments were applied in terms of the number of trees retained. So not in terms of the basal area retained, because that would automatically give a good result, presumably, but instead it was in terms of the number of trees retained. The points and the solid lines show the actual measurements, the observations, and the dotted lines show the predictions. And this was repeated for 23 different plots. And there were different combinations of species and different sizes. So the different colors here show the different species, and the different sizes of the circles show the different size classes. And these graphs showing the predicted versus the observed biomass indicate that the model was working relatively well. So then it was used to try to understand why these types of forests have been growing faster over the last few decades. So it seemed to be a combination of changes in management and changes in climate that were causing that. And we were able to use the model to split this up a little bit. And lastly, I mentioned that we decided to use stand level models instead of tree level models. And one of the main reasons was that we couldn't find improvement in predictions by using tree level models, except when those models were very detailed process-based models that were set up specifically for a certain site and a certain combination of species. And this was based on literature reviews and our own work. And we were surprised to find that stand level models were as accurate at tree level models in these relatively structurally complex stands, or sometimes they were even more accurate. And it looks like there might be 2 main reasons why this was happening. One of them is that there are lots of nonlinear relationships at the tree level or at the leaf level that actually become linear at the stand level. So just as an example, this is a relationship between transpiration and photosynthesis, and this is at the level of leaves in seconds, and it's nonlinear. This is the same relationship, but this is at the level of the stand and months, and this is linear. And the same contrasting shapes occur for relationships between light absorption and productivity, or between leaf area and light absorption. And the reasons why are described in this paper listed down the bottom by Binkley et al. But the point is that the linear relationships at the stand level can simplify the model, and they also simplify the model interpretations. The processes that I listed earlier in some of the other slides can generally be summarized at the stand level, and often they're summarized even using linear relationships. And the second reason why it appears as though the stand level models were as good as the tree level models was that the tree level models often use stand level inputs for some of the most important factors that influence growth. For example, for the soil resources. But the soil resource availability is usually very heterogeneous, and this is what's shown in this graph. On the left is a map of the spatial distribution of soil water availability in some spruce and beech mixtures in Austria. Then on the right is a map of the leaf litter-form mass in another plot. And as is often the case, there's a lot of spatial variability within these plots. And a problem with tree level models is that even if they take account of the spatial positioning of the trees, or if they use competition indices or something similar, this still doesn't really account for this variability in the soil water and soil nutrients. And these are 2 of the most important factors that influence growth. So to conclude, there's quite a dynamic spatial and temporal variability for species interactions, but it appears as though relatively simple stand-level process-based models like 3PG can actually reproduce these interactions between species and between size cohorts. And when we were doing this work, we found that it was quite important to select a model that focuses on the main species interactions. It doesn't have to do all the interactions, just the ones that are having the biggest effect. It's also important that each of the different submodels of the model are validated carefully. It doesn't seem to be enough just to validate the stand growth or the stand stocks. It's important to validate the actual processes as well. Thanks.
Thank you so much. Before we move on to question and answers period, let me share my screen real quick to just draw your attention. Oh, wrong screen. Nope, just the wrong file. Draw your attention to the upcoming webinar in May, where Dr. Mauro from Spain and Dr. Frank from the BLM in the United States are going to talk about small area estimation in forest inventories. And then the October webinar is lined up already, and Dr. Stella Britt from Aqua will talk about the assessment of deforestation and forest degradation in community resource management areas in the northern savanna zone. If you do have any suggestions for other speakers, I'm still looking for speakers for September and November this year, and to keep to learning about upcoming webinars, you can subscribe to the Division 4 mailing list, and I typically send out the announcements there. And also, if you check the IUFRO calendar, the announcements will be posted there as well. With that, thank you very much for your attention. Please stay on for a Q&A session with Dr. Forrester, and I will stop the recording now.